Hello, I'm going to do a quick discussion on homeostasis. This is our first lecture for human physiology, and it covers some bits in chapters 1 through 73, and I also would like you to review chapter 2. We have a lot to cover in this lecture. I'll start out with some simple terms that we are going to use throughout the semester. I'll also tell you the difference between physiology and pathophysiology, and then we'll jump into an example of homeostasis by talking about thermoregulation. Physiology is, quite honestly, a little bit boring. It's the science that is concerned with the function of the living organism and its parts, and of the physical and chemical processes involved. Basically, it's how stuff works in your body. And again, that's a little bit boring because most of you are in my class because you want to help people who are sick. Really, the cool factor is the pathophysiology, the study of disordered body function, that is, disease. And it's the basis for clinical medicine. It's the reason why most of you are in my class. So I will teach you physiology, but I will always relate it back to the clinical manifestations when someone is in a disease state. In other words, you're going to have a lot of pathophysiology in this class. A very much recurrent theme in our class will be homeostasis, or the maintenance of a stable milieu intérieur, as coined by Claude Bernard, a very famous French physiologist. So milieu intérieur means a stable internal environment, and it was Claude Bernard who was actually astute enough to recognize that that meant maintaining the plasma. And that's really important that we understand that his focus was on the plasma. And if you think about it, plasma has a lot of stuff dissolved in it. It has our oxygen, it has CO2, it helps transport formed elements like red blood cells that also carry oxygen and CO2 for us. Plasma brings nutrients to all the tissues of our bodies and also removes waste. So he was quite astute when he was referring to the plasma for maintaining a stable internal environment. It's all about the plasma. He also recognized that if the pH in the plasma changed drastically, or the salt content changed drastically, or temperature changed drastically, that this could lead to the denaturation of proteins. We don't want that to happen. That's why we need a stable internal environment to prevent denaturation of proteins. We need to keep the cells under an optimal condition so that they can function and survive. Proteins are the workhorse of the cell. You lose your protein function, you lose the function of the cell. So again, it's all about the plasma, maintaining the plasma in a very stable internal environment, stable environment, so that we do not denature proteins. Key terms that we will be using frequently throughout the semester include variable. Variable is anything we measure, anything we can measure. We take temperatures from our patients all the time. We do blood pressure recordings. We do a full assessment, right, of their vitals. That's what we say, take their vitals. We take their pulse rate. We can even collect urine. We can collect urine over time. We draw blood, and we get a huge amount of data from a patient's blood. We get their pH, we get their sodium concentration, shown here on the screen, their plasma concentration of glucose and other things. So a variable is anything we can measure. All variables need to have a set point. Now, set point, that term, is very misleading to a lot of students. They think it's just one number. Set point is not one number. Set point can be an acceptable range, and we usually base this acceptable range based on the population. For example, if I were to take 100 students and evaluate or record their heart rate, for example, I might get a range where most of you fall into. And we can do statistics on that, of course. So this set point is really an acceptable range based on measures taken from a certain number of the population. Throughout the semester, I will be giving you many set points to many variables. And sometimes I will say, I want just one number. For example, I can give you an example right now. pH. An acceptable pH in the body is 7.35 to 7.45. But for our class, we're going to hold to a very hard 7.40. And there's a reason for that. It's really to eliminate any controversy during an exam. So that would be an example where I'm going to say, here's a range, but I want us to stick to 7.40. There will be others, and I will alert you to them as we go through the semester. A deviation is a change from set point. If the patient's variable migrates out of an acceptable range, we say that's a deviation. For example, pH. Acceptable range, 7.35 to 7.45. If the person deviates to 7.46, that's out of the acceptable range. That's a deviation. Correction is really how the patient will compensate. Their body will have an internal compensatory reaction where they're trying to get the pH, or the body will try to get the pH back down to the acceptable range. How much compensation depends on how great the deviation is. If the person has a huge deviation, then the compensatory mechanisms will be launched in force. The closer you are to set point, the lower the compensatory mechanisms. Here's my example. If I am at work and I want to go home, I'm going to take the freeway. And on the freeway, I'm going to be going at a very great speed, right? Speed limit is 65 miles per hour on most freeways. But as I exit the freeway onto surface streets, clearly I shouldn't be going 65 miles an hour. Surface streets by my house are closer to 45 miles per hour. And as I enter into my housing subdivision, clearly I shouldn't be going 45 miles per hour through the subdivision streets where kids are playing. The point is I'm getting closer to my home. That's my set point. And the closer I get, the less deviated I am. If I'm at work, I'm really far away from my set point, my home. That's where I want to be. The closer I get, I should slow down my speed. When I pull into my driveway of my home, clearly I should not be going Dukes of Hazzards and just press on the accelerator and slam through the garage door and into the downstairs bedroom. So the closer you get to your set point, the more the compensatory mechanism should alleviate. You don't want to overshoot your set point, right? You don't want to go hardcore and swing hard in the other direction and deviate in the other way. So, correction, the more removed you are from set point, the greater the deviation, the greater the compensatory mechanism. The closer you get to your set point, the less the compensatory mechanism will be. There are two ways that we have studied compensatory mechanisms. There is a thing called negative feedback and positive feedback. Negative feedback promotes stability. Most of the deviations that occur in a human body away from set point, they will be corrected through negative feedback corrective mechanisms. This means that when the variable deviates from set point, there will be some sort of way that the body detects this deviation. There has to be a sensor, something that senses the deviation, that's monitoring it. then this signal is compared to the set point. And this is usually in an integration center like your brain and spinal cord. Brain and spinal cord know the set points, usually the hypothalamus for most of the set points that we're talking about, but not all. So the brain, spinal cord evaluates what the set point is, evaluates the input signal from the sensor, and it creates a different signal, the deviation. How far deviated are we? Then there is a pre-programmed correction that's triggered. That's gene expression, and this is the physiology that you're here to study, how the correction occurs. This is a huge, huge undertaking to understand how a system corrects itself or tries to correct itself. Then the brain or spinal cord, the integration center, has to release an output signal. And this output signal is going to target an effector, something that can perform an action to help the body get back towards set point. And the ultimate relationship is in this negative feedback correction is that the correction is opposite in direction to the deviation, shown down here at the bottom of my PowerPoint. If your body temperature elevates too much, for example, well, then the correction is to try and cool you back down. That's what I mean by opposite direction. And the variable returns or returns toward the set point. We are never really at our set point. We're never locked in. So we're trying to return toward set point, not to set point. This is very different compared to positive feedback corrective mechanisms. Usually with positive feedback corrective mechanisms, there is a huge, big change. And we call it an avalanche experience, where deviation leads to more correction, but in the same direction as the deviation. And that's really weird. So we say more gets more gets more. It promotes a change in one direction. And if it gets out of control, this can lead to an unstable environment. It leads to instability, as I just said, and it really is the underlying cause of most diseased states. So positive feedback mechanisms are not used robustly in the body because they have this tendency to get away from us and can lead to a diseased state. Some good examples are childbirth, where the woman shown here is terminally pregnant with a parasite inside of her. I know, I call it a parasite. And as that fetus gets bigger and bigger, it pushes on the cervix. Well, the cervix, it will send signals up to the brain, most notably the hypothalamus. And the hypothalamus, as the cervix is being pushed on by the fetus's head, will release oxytocin. Well, oxytocin causes uterine contractions. So the uterine contractions will push on the fetus, and the fetus's head is going to push even more on the cervix. More pressure on the cervix leads to more oxytocin, more uterine contraction. More pushing of the fetus head on the cervix leads to more signal to the hypothalamus. You're hearing more leads to more leads to more. And the more that the cervix is pushed upon and stimulated, the more it starts to dilate. And this will continue until the cervix is dilated enough where the fetus can be born. And then, of course, this labor has to stop once the baby is delivered. So all positive feedback, let me say it again, all positive feedback mechanisms must ultimately be stopped with a negative feedback mechanism. You have to stop that avalanche effect. Another example of this we'll learn in Unit 2 is an action potential generated by a neuron. Another example that we'll learn of positive feedback where more leads to more leads to more is blood clotting, or hemostasis as we call it. And then another example of positive feedback back in a disease state that is covered in the readings for this PowerPoint is heart failure. And this is covered in those early chapters that I showed you on the first slide.